Okay, welcome everybody. Before I jump into this stuff, I did receive a message in the chat just a second ago questioning some pack back makeup stuff. So the deal with pack back, there are 14 weeks worth of assignments that are going to be posted or that you're going to have, but I'm only going to count 10 of those. So there's a little bit of wiggle room. The extra 4 will be extra credit. If you've missed some pack back, you can, on the current week, you can't go back to previous stuff, but on the current week or the following week, whatever it might be, you can do some extra questions. The total amount of responses is what I'm looking at, all right? Not all 14 weeks worth of things, whatever, all right? So yes, you can make some stuff up, but you can't go back. You can't jump back to previous weeks because once that stuff closes, it's closed. But you can do some more, all right? Or you can go with the 10 and be all good to go, all right? With that said, midterm grades, it's midterm week. As I submit midterm grades, I'm not going to take into account the individual pack back scores. I'm going to look at all of the pack back, and if there are people that have done 0 on the pack back, you will know that because I will incorporate that in there. But if you have attempted some pack backs, if you've done a few at least, I'm not going to take any of that stuff into account when I deal with your grade. Last go around, I didn't do any pack back stuff at midterms. And then at the tail end, I realized there were a few people that signed up for pack back, and that's all they ever did. So it could be in your best interest to know if you are in that boat. And so I will deal with it that way. But otherwise, I'm not merging all of the stuff. It's not going to go into the grade book on Moodle. None of that stuff's going to happen. Is that good? Any other questions? Any questions from my Zoomers? I mean, that was a question from there in the first place. Okay. So we talked about this slide. I just opened it up right to this point. I'm not going to go through the details with this again. I do want to just mention, truly, there is a tremendous amount of complexity with the process of DNA replication. And even though this looks very detailed, we really glossed over quite a bit of stuff, just trying to simplify it and show some of the main ideas, all right? Quickly, there are leading and lagging strands of the DNA, anti-parallel, all right? And you can see 3 to 5, 5 to 3 as we are moving away in opposite, right? And the leading strand is a continuous synthesis of those nucleotides as the polymerase is moving in the proper orientation, moving toward the replication fork. And in turn, as helicase is unzipping, polymerase just keeps booking right along. On the lagging strand, it's the opposite. We're going in reverse. And in turn, multiple, we don't see the individual RNA primer up here at the top. One primer was laid down to start polymerase up above, but down below with multiple discontinuous sequences, every one of those needs to be primed in order for polymerase to get on there and start moving. That makes it so there's a bunch of green bits, RNA molecules, that get laid down to allow polymerase to come in. So other enzymes, DNA polymerase 1, this is polymerase 3 that's doing the initial lay down of the nucleotides. Polymerase 1 does some proofreading, and it goes through and recognizes the RNA primers, chewing them off and putting down DNA nucleotides in its place. So there's a lot of that stuff happening on the lagging strand. And there's another enzyme called ligase that helps to fuse the adjacent Okazaki fragments together, building the covalent bond in between those respective sugar phosphates that aren't linked together initially. That's the idea, all right? So in turn, one parental and one new strand laid down on both, all right? There are, you know, as we said as we were starting this, there's differences when we make comparisons between prokaryotic and eukaryotic replication. There are similar players when we think about all of the respective enzymes that go in there and untwist and unzip and stabilize the polymerases, all of these things similar, all right? There are definitely some differences when we think about different types of microbes, as we mentioned last time, with like those thermophiles or the hyperthermophiles that are living in boiling or near boiling temperatures. Their proteins or their enzymes are very different when we think about our own, with the fact that they can tolerate extreme temperatures, all right? And that opens up the idea really for the next chapters, think of some biotechnology and how we may manipulate that, all right? So in eukaryotes, linear chromosomes, it says typically linear, and as I was reviewing this, I was like, I can't really think of any that aren't linear, all right? I can think of inside of mitochondria with circular chromosomes, but that's a different story in and of itself, all right? In the nucleus of eukaryotic cells, linear chromosomes, right? There's definitely potentials for variation when we think about the numbers of chromosomes. Usually in a prokaryote, a single circular chromosome, and in eukaryotes, many linear chromosomes, all right? So it's a little bit different. And in the prokaryotes, and we'll see an image of them in just a second with their circular chromosomes, how we go about replication, slightly different than in the eukaryotes. So where we start, like kind of where that zipper would start unzipping, we call an origin of replication. And this is where we get the ability to unzip and start stabilizing, and the helicase, the zipper bit, starts breaking those hydrogen bonds in between the nucleotides and going in both directions. That's the same with the prokaryotes, both directions, but not multiple origins of replication. That's what we see in eukaryotes. Many points in there that allow it to start, all right? This lends to opportunities for the DNA to get replicated really pretty quickly because we've got multiple origins, and for these processes really to be going, as we see as we move down simultaneously, moving toward one another, all right? And as we showed in this previous image, I'm backing it up to it, we can see one part from going from the origin of replication in the proper orientation is the leading, and on the same strand going in the opposite orientation is lagging. So you get both. It's 50-50. Half of it's leading, half of it's lagging, and the opposite on the anti-parallel strand. So same idea, we're just not showing those details, all right? And once it's all said and done, we've got two copies, one side parental and the other the new laid down in red, all right? And then we would go through, after we've gone through our synthesis phase, what we're seeing right here, then we would move into mitosis or meiosis and separate them into respective individual cells, all right? We're not going through those details right now, all right? So the fact that the eukaryotic chromosomes are linear, there's also some differences with them that we don't see in the circular prokaryotic chromosomes, right? Telomeres are big, long strand or sequences, not strands, big, long pieces of DNA that are on the ends of the chromosomes, both ends, right? And they serve really kind of as a sort of protection for the chromosome. And we can see here with some enzymes, some telomerase activity, how we can maintain these chromosomes. This is really with the telomerase activity, something that we would think of more maybe in like cancerous cells, where we've got high levels of telomerase activity. Normally, we're not thinking about this functioning in typical cells. As they go through their rounds of replication, those sequences that are being laid down, there's a little bit of overhang that we can see on one side of those chromosomes where the RNA polymerase isn't capable of linking up to lay down stuff for DNA to get put down there in the first place. And that little overhang gets broken off. There's enough telomerase on the ends of these chromosomes to lend to about 70 or 80 rounds of replication. Kind of depends on the specifics, but about that many, right? So that really kind of governs the longevity of that specific cell line. They've got the potential to divide almost 100 times, all right? And once we get through about that many divisions, all of the sudden, what's breaking off isn't just this repeated telomere DNA, it's the actual business. It's the genes that those chromosomes are carrying that start getting impacted. And then that cell line will die off, all right? So the telomerase has the potential, this enzyme, to lay down new material, right? A fountain of youth, potentially, right? Cancerous cells, for sure, right? Cells that don't know how to die, right? Because every time they replicate, they feel like they're brand new little baby cells, all right? Fresh and just starting, right? So that's the idea with that. All right, so this is bacterial replication, circular chromosome, and one origin of replication, the DNA basically gets opened up, if you will. And the new stuff laying down in red as we are moving through there and unzipping the DNA, we get another, basically a double loop. And we start, although we're showing this separated, they replicate and then we nick them and separate, all right? So this shows the synthesis of the second strand, but really it's kind of happening simultaneously with both strands getting laid down, all right? One on the outer, the other on the inner, all right, is how it actually would be going. And that's what we're showing here with the red on the outside or on the inside, all right? And bacteria have potentials. Again, we've mentioned this repeatedly, depends on the type of bacteria, but they may be able to replicate every 20 minutes or so. So the process of synthesizing its nucleotides, if it's got ample nutrients, there's plenty of ingredients to do this stuff. The process of DNA replication is very rapid, all right? Much quicker than happening in the eukaryotic cells, but that gets kind of a leg up with the multiple origins of replication to speed that process up. But it's still quicker with these guys. It's much smaller chromosome in the first place. So shifting over from DNA replication, taking the central dogma that we started this chapter with, DNA to RNA to protein, that's where we want to transition. And we want to think initially about some transcription. There are definitely, we'll make some comparisons with these guys in a second as well. There's differences when we compare prokaryotes to eukaryotes. It's the same idea. We want to make messenger RNA, but there are some variations that we'll touch on, all right? So it's not the same as that RNA primer that's laid down that allows the polymerase to recognize and start going, but it's a similar sort of an idea, all right? This promoter region is like that, and it's the spot that is going to allow polymerase, RNA polymerase, to recognize this piece of DNA and basically say, this is where I link up, all right? You would like that, a cell would like that to happen before the details of what we're trying to make in the first place, right? The polymerase needs to get on there and then start doing its job, right? Otherwise, it would link up and likely miss some of the material that it should be transcribing, right? So the promoter is just a little bit upstream of the information that we want to make a copy of, all right? So as we look at this, our DNA, again, anti-parallel, all right? We're looking at this strand of DNA, and we can see this bit 3 to 5 prime. And as we are making our messenger RNA that's in pink, it's linking up in the 5 to 3 prime direction. That is the way that RNA, messenger RNA molecules are synthesized, all right? So that comes in like this, the direction of synthesis, the polymerase bringing in the RNA nucleotides, and if they complementary pair with the DNA sequence below, they're allowed to fuse that backbone, sugar phosphate backbone, and now we've got our messenger RNA, all right? So the template strand is the strand that's being read, and we're making messenger RNA off of, all right? The strand above is the non-template strand, right? It technically is a mirror image of this message, right? Although we've put uracils in the place of thymines, it looks exactly the same, right? As we compare this one says, I want you to build it, but really, this is what we're building, right? And the message says, okay, I can do that, right? The transfer RNAs that are gonna be bringing in amino acids, we'll see them here in 1 second. They have a portion on the end of the transfer RNA that complements the sequence that looks the same as our template strand. And it's bringing in the amino acid. So this says, I want this. The message says, okay, and the tRNA brings it in, all right? That's the idea. Transcription and translation will happen in just 1 second, all right? So this figure is looking at the RNA molecule, the messenger RNA. The DNA would have those instructions, and now we've made the message, all right? I assume most of us have seen a table like this before. Hopefully you've got a bit of an understanding about some of this stuff. I think it's kind of interesting. So there are, with a number of these amino acids, 3 letters, this codon, 3-letter word, if you will, codes for a respective amino acid. Some of them, there's only one way, but most of them, there's multiple ways to get the same amino acid. That becomes pretty important because you could have some subtle change in the genetic code, some, little bit of variation, and maybe that variation doesn't matter, because if it ends up saying, bring in this same amino acid, who really cares? You're bringing in the right stuff, all right? But if it says something different, and now we bring in an incorrect amino acid, now we've got problems. And we'll talk about some mutations, deviations from this table that set the stage for problems. Mention that shortly, all right? So the way we utilize this table, the first base in our codon is on this side, a uracil, a cytosine, adenine, or a guanine. Our second position going across the top, and the third position, an individual row with each of these. All right. So first square up here, the first letter is a U, so uracil in the first position. The second one is a uracil, and as we see here, they're all uracils going down, right? The first bit up here, all uracils going across, right? And then the third position is one of each, right? A uracil, a cytosine, an adenine, or a guanine, right? And now looking at this, this first bit, we can see a phenylalanine or a leucine that are two of the amino acids that, based off of these three-letter codes, we would be creating, right? So with this first square here, two different potentials for amino acids based off of what we've spelled out. If we move right down below, there's another one that is all leucine, 4 ways to make leucine right here. And in this instance, it doesn't matter what that third position is, as long as you have a cytosine and a uracil in position 1 and 2, whatever you have in the third position, one of those 4, we're bringing in leucine, all right? We call that redundancy. More than one way to make or to build this protein sequence, all right? It's all coding for the same thing. And in some species, bacteria, good examples, we're in microbiology, there are numerous types of bacteria that we can start diving deeper into and looking at genetic sequences and recognize some stuff that we call codon usage bias, where a type of bacterium might prefer, CUC over CUG. And almost all of the sequence in this respective bacterium would have that as its specific codon for leucine. And a completely different one, maybe E. coli has CUC, and maybe Shigella says CUG, right? Both gram negatives, both intestinal organisms, right? Some similarities, but some subtle differences. And maybe looking at a little of that and understanding, not that I expect we understand that in here, but understanding a little bit of that might clue you in that you're looking more at this versus that, just seeing sequence data, right? Which is kind of interesting, right? So in green over here, the AUG, this is start codon. All of our protein sequences begin with methionine or with AUG as the start codon, all right? But if we were looking at a sequence of DNA, just because we see the complement of that, right, doesn't mean that that's a start codon, right? It could be, but maybe not. It might just be coding for methionine, right? And trying to understand the, specific genes when we're doing sequencing can be very difficult when we're thinking about this. Where is the start codon, all right? And if you're looking at a bacterium or you're looking at a eukaryote, there's definitely some differences with some of that, all right? And you ultimately get a lot of practice going through that, really trying to evaluate where it's actually starting, all right? Over here in red, 3 different stop codons, all right? And I like to think of this stuff like a children's book, right? Like little kid book, 3-letter words, right? We're just learning how to read stuff, right? 3-letter words are what we're looking at. And these are the codons, coding for all of these things, right? And if you're writing a sentence, you're going to capitalize that first letter in that sentence. The start of the sentence is this, right? Methionine, it's our capital letter, right? And technically, if we're doing some grammar up in here, if you're writing a sentence, there's a handful of ways you might punctuate that sentence, right? You might put a period, a question mark, or an exclamation point, 3 different punctuations, right? And in turn, 3 different stop codons. It's brilliant. It makes perfect sense, right? We're saying something, right? Whether it's a statement, a question, or an exclamation, build me some damn protein, right? That's what I want. So multiple ways potentially to bring that in. 1 way to start it and 3 different ways to stop it, right? With that, on my little dots right here, tryptophan over here, our friend in Turkey that they say makes you sleepy. It's like methionine where there's only a single codon that codes for tryptophan, right? But pretty much everybody else on here has more than 1 way, right? Just kind of depends, right? So we're going to jump over and talk about transfer RNAs that are bringing in these amino acids, but I want to leave it on this table for 1 second, all right? As we are building transfer RNAs, this is RNA molecules that we're coding for in the sequence of DNA, right? We're not going to make every single one of these. We're not going to make an individual transfer RNA, right? That's 61 different transfer RNAs. That's a lot of stuff that technically you really don't need, right? I'm going back to leucine because I always find out, you know, I'm going to switch it. We'll move over here to arginine, all right? Which I have been told that's a pirate's favorite amino acid. That's a dad joke for you, right? Arginine, right? Arginine over here, we've got 1 transfer RNA that brings in arginine. It doesn't care if this 3rd position is any of these bases. It's going to bring it in. And we say those tRNAs, they wobble. They can wobble on the back end, the 3rd letter, but the 1st and 2nd, there's no wobble. It's got to be specific, all right? So 1 transfer RNA bringing in this arginine. But as we look down below, and I quickly glanced before because I was like, oh crap, is there only 4 ways for arginine? No, there's another one. Down below, there's another way we could make arginine, all right? But in this case, adenine's in the 1st position, and then we've got a guanine in the 2nd, and then adenines or guanines in the 3rd position. Different transfer RNA for these guys because of the front end, right? We don't wobble on the front, all right? So this minimizes the amount of transfer RNAs that a cell needs to produce, right? We don't need to make 61 of them with this redundancy, all right? But we do need to make an extra one for arginine down in this box below, right? There are, I assume you remember some of this with the nucleotides. There's some differences, and these are ring structures, right? There are purines and there are pyrimidines, right? Adenine and guanine, those 2 nucleotides are purines, right? And the uracil, the cytosine and guanine, cytosine and thymine, if we're talking DNA, not guanine, that's down below. Those are pyrimidines, right? So in this case, it doesn't care as long as it's a purine, right? It brings it in, right? And up here, pyrimidine, it brings in the serine, right? But over here, it doesn't care what that position is, right? In that 3rd, or what that base is in the 3rd position. So different tRNAs in these instances, but not 6, all right? Dramatically cutting down on that. So 1 difference with our prokaryotes, I mentioned before, in the genetic code, in the DNA, it's all business. It's all coding, all right? So once we start reading and building messenger RNA, we can create multiple messages. And as we are making these messages, we can start translating them and building proteins simultaneously. And you don't see that happening in eukaryotes, right? There's got to be some processing that happens. Eukaryotes, in their DNA, we see introns and exons. The exons are the business, and the introns, they're the in-betweens. They're the garbage or the junk DNA that's not truly junk, all right? Little spacers, chunks of information that's not spelling out this is part of the gene, right? But potentially helping for that protein to be able to fold up properly as we ensure that we're copying it correctly, all right? So once we've done that in the nucleus, we're going to process it, chopping out the introns, adding some extra packaging to it, and then exporting them out of the nucleus. Transcription and translation happen in different parts of the cell, and they don't happen simultaneously when we're talking eukaryotes, right? But in prokaryotes, it does, right? And it's like mass production, assembly line sort of an idea, all right? So here we can see our RNA polymerase. We're reading our DNA, we're making messenger RNA, right? And in turn, this is not DNA, right? As I'm looking at this, I'm like, what the heck are they trying to tell you? These little circles come on off of here. These are the amino acids, right? This would be protein that a protein that we're starting to build, all right? So little amino acids, circles linking up as our ribosomes are reading the messenger RNA. This bigger bit right on here is the ribosome, all right? And those are free floating in the cytoplasm of a bacterial cell, all right? That's what we see here. Polyribosomes, numerous ribosomes, and they start linking up to the message before we're ever done making it, all right? Right as soon as we've got enough space for a ribosome to bind, we start building protein, all right? And multiple ones can link up, all right? That's the idea as we're booking along this sequence of DNA. So hopefully that makes a little bit of sense. So our transfer RNAs or tRNAs, these guys are coming into the ribosomal complex. The ribosomes sandwich around the messenger RNA, and the transfer RNAs move into these ribosomes. And if they can complementary pair, the anticodon is the part of the transfer RNA that is looking, if it looks, it's looking at the codon on the messenger RNA. And if they are complementary, there's a brief bit of hydrogen bonding that occurs between these two, and that's enough for it to say, we're in the right position. And there's an amino acid that's stuck to the other end of this transfer RNA. We don't see it on here, but it would be linked up right here. And this amino acid in that ribosomal complex gets donated to the growing polypeptide chain, all right? And we just keep bumping it along as we move through there. So with the transfer RNA, there's really kind of two business ends to it. One that's reading the message and the other that's bringing in that amino acid and donating it to the growing chain, all right? Sometimes, as we look at this, they call this the second code because it's reading this other message, all right? Bringing in the proteins, all right? So that's what we're seeing here with these circles being those amino acids, not, sorry, wrong way, not DNA, all right? Because that's wrong. Thanks, OpenStax. Okay. That's okay. I'm curious if that figure is still that way in the actual text or if it's been adjusted. I don't know. I'm not going to open it up right now to look, but hopefully in the text, it's been modified. All right, anybody have the book open right now? I thought not. Okay, what? There's a book? Okay, I know you know that. So here's our bit of translation. And like I said, in a prokaryote, this is happening simultaneously. Once we've got the first bit of messenger RNA laid down, we can technically start this process. All right. So before all of this starts, we've got the pieces free floating, if you will. The message, the ribosomes exist before they're doing the actual reading, if you will. They exist as separate subunits, all right? So initially, we have to start this whole process, and it happens with this initiation, all right? So there's a complex that forms. As all of this is still separate, our tRNA with the anticodon that looks at our start codon right here, our tRNA links up with this. This is like the catalyst that says ribosomes unite. And in turn, they sandwich around this tRNA that has linked up to the message. And this is the initiation. The small and large ribosomes link up around the message that already has that first transfer RNA on it, right? And we call this a ribosomal complex once it's all linked up together, right? At this point, the ribosome starts going down the train tracks, if you will, down the messenger RNA, right? And as it's moving through there are really three positions inside of these ribosomes, which are basically a big enzyme that's helping to catalyze these reactions of amino acid chains, right? Growing, building proteins. So we bring in a amino acid or bring in a transfer RNA with its amino acid. If it pairs, we move those growing protein chains to it, fusing them together, all right? Catalyzing a covalent bond from one amino acid to the next, all right? And we call those covalent bonds peptide bonds, all right? It's a covalent bond making proteins, all right? So that's all happening in this ribosomal complex, all right? Fusing them together. And once we've donated this, the one that was in the middle now gets to the last position. It's the ejection seat, basically kicking it out. And this amino acid that's removed from the ribosome is decharged, if you will. It doesn't have its amino acid on it anymore. And it links up with an enzyme that will put the proper amino acid onto that amino acid attachment site, right? And it's ready to come back into the mix, bringing in its respective amino acid, right? And this happens, we progress along the message until we get to a sequence that basically doesn't code for any amino acids. That's the stop codon. A release factor, it's basically the punctuation, and the ribosomes disassociate, go back to their free-floating separate bits to potentially flip right back around and start over, dependent upon the situation. So this compares the two, all right? The prokaryotes we've already really been describing, where all of this starts as soon as the first little hint of message has been generated, all right? So transcription and translation really occurring simultaneously, and the potential for multiple ribosomes to link up and form this polyribosomal complex to be pumping out countless proteins starts happening almost the instant that we've started making message, right? And again, all of this stuff is just in the cytoplasm. In our eukaryotes, I mentioned this the littlest bit. it's different, right? There's other material in there that's providing some spacers between these respective genes, the introns, the spacers, right? So we make this sequence as we read the messenger or read the DNA and make this messenger RNA, and it's got intron and exon material together. We do a little bit of processing inside of the nucleus, and we remove those introns, sandwiching the exons together as we chop out the introns, which in turn gets degraded and reused. It's nucleotides that we can keep building more message with, all right? So we process it that way. Initially, we call it pre-mRNA because it's got extra information in there, all right? And then we get rid of it and we put a cap and a tail on it that helps to facilitate its movement out of the nucleus and its docking with ribosomes, whether they be free-floating ribosomes in the cytoplasm or ribosomes that are associated with the rough endoplasmic reticula. Really depends on the fate of that protein and some of the business that we capped or tailed it with helps to get it to go there, right? The little tail is like a zip code that you put on a letter that you're going to mail to somebody, right? If you don't put the proper zip code, it's not going where it needs to go, right? And that's really what it's kind of helping to do, all right? So as we've got this image right here, all right, a little segue to our biotechnology, all right? When we think about building human stuff in a bacterium, we need to go through a couple of extra steps because you can't just take the DNA from a person and chop out that gene and say, sweet, here's the gene for insulin. I'm going to just pop it right into E. coli and have him pump that out, right? It's not going to work like that because there's a bunch of extra stuff, right? All of those introns. You can't deal, the bacteria can't deal with that. It doesn't process messenger RNA. And now what you'd be trying to create will be a bunch of garbage, right? All of those introns, it's not coding for anything, right? So in order to make our bacteria build human stuff, we have to do this first, and we have to process that DNA, chopping out all of the introns. And now we've got mature RNA, but you can't take RNA and pop it into DNA. That doesn't work, right? So we've got to take another step. We've got to process it, make it so it's mature, and then we use a new enzyme called reverse transcriptase. And that enzyme reads RNA and builds DNA in its place. So you can take a single strand RNA copy and build double-stranded DNA. We call it cDNA or complementary DNA. That message says, I want you to build this. We run reverse transcriptase through it, and now you got a chunk of DNA that says, here's my instructions, coding and non-coding, right? And that bit you can pop into a bacterium, and now it's got everything it needs, and it can read it and say, oh, I should build insulin, and I don't care what it came out of, I'm going to build it, right? And there you go, all right? Now we don't even really need to cover the next chapter, right? I did it all in one quick slide, all right? I think that's pretty interesting, all right? And really opens up the potentials for all sorts of opportunities to genetically engineer all sorts of things. There are other opportunities where maybe you don't want it in a bacterium, all right? We could manipulate stuff. We could take a gene. We don't need to do anything with the RNA. We could take a gene out of us. We could pop it into a yeast cell. Saccharomyces is a great one, right? And yeast cells are eukaryotic. We're eukaryotic. They've got all of the materials needed to process and remove introns. So they could take human genes in a yeast cell and build human proteins. without any of the steps that I just described that you'd need to do to pop them into a bacterium. Just depends on what you're truly trying to do and how you're trying to, what you're trying to produce, all right? That sets the stage for some of those details. All right. So quick comparison about our translation between our bacteria and eukaryotes. The ribosomes, they're the same but different, all right? In eukaryotes, they're bigger, right? But basically it's the same sort of an idea, separate subunits sandwiching around and allowing the tRNAs to come in, all right? But different sizes, right? Same idea. The amino acids that start codons, it's methionine, but in bacteria, it's slightly modified, the F in front of it for a formal, not formal like a dance, all right? Like a methyl group, all right, that's on the methionine. That's the formal. And then the Shine-Dalgarno sequences present in our mRNA. We see that in our bacteria, but we don't see that in the eukaryotes, and this ultimately lending to the transcription and translation simultaneously. We don't need to go into details with this, all right? I skipped over that before in the first place, all right? And as I said, this happening simultaneously, whereas in eukaryotes, you got to process first, all right? I'm going to stop right there. As I get to mutations, I think this is the perfect point to pick up on Friday. And I'm pretty sure you've probably had enough, right? Probably so. Did I lose you again at helicase? Right as I started off, you're like, dang it.
No, I stuck with it.
You stuck with it. Okay. All good. Any questions for the home viewers? I see them dropping. They're dropping like flies. Okay. Fair.